Everyone, hope everyone is doing good. Today my topic of discussion is poliomyelitis essay, which can be asked in microbiology university examinations. Let's get started. Okay, poliomyelitis. What is poliomyelitis? Poliomyelitis is highly infectious childhood disease, which is also known as polio. The causative organism of polio, obviously everyone know, that is polio virus. Okay, what polio virus causes in our body? It causes acute flaccid paralysis, that is due to the involvement of nervous system. It is also in verge of eradication. Okay, let's move into the morphology part. Okay, this is the picture I drew here. Is the polio virus external structure. Okay, this one is VP1 and this one is VP2. VP is viral, VP2 and VP3 and VP4. The inner one is VP4. The type is enteroviruses. The family is Picornaviridae family. It is non-enveloped. You guys know it is non-enveloped. And shape is spherical in shape. And symmetry is icosahedral symmetry. And capsid is 60 subunits with 4 viral protein. VP is viral protein, which is of 4 in number, except pyricovirus, that is 3 subunits. It has single-stranded positive sense linear RNA. This is single, right? Single-stranded positive sense linear RNA. The antigenic type. Poliovirus is of 2 types, that is wild poliovirus and vaccine-derived poliovirus. The wild poliovirus is natural disease-causing agent. The vaccine-derived poliovirus is due to the vaccine strain. It regains neurovirulence property. This is very important. We injected the vaccine into the person. It's got reactivated. The reactivation is neurovirulence. And it causes disease in human. Okay, let's talk about detail of wild poliovirus. Wild poliovirus is of 3 types: WPV1, WPV2, WPV3. That is type 1, type 2, type 3. All are WPV1 is natural cause, and these 2 guys are globally eradicated. All 3 are antibody response specific, and there is no cross protective, which is very important. Okay, how the polio virus causes disease in our body. Let's start the pathogenesis here. The transmission is due to fecal-oral route and respiratory deposits and conjunctival contact. Okay, this is lung, this is eye, and this is fecal-oral route, which multiplies in tonsil and submucosal lymphoidal tissue and the intestinal epithelial cells and paste patches. First, let me draw a picture. Okay, this is human. Here tonsil is there, right? The polio enters the tonsil, lymphatic tissue, intestine. In intestine, you people know pair patches is there. This rough diagram, okay. And receptor is this is very important neat PG question. CD155 receptor. Okay, how the poliovirus spread to the CNS and spinal cord? It is due to 2 mechanisms: hematogenous spread and neural spread. And the hematogenous spread is most common. That is, it first spread to the regional lymph nodes and causes primary viremia and multiply within the reticuloendothelial system and causes secondary viremia, and ultimately it's go into the brain and spinal cord. Okay, this is one method. The other method is, first it spread to the neurons and the tonsil, and in the tonsil there is glossopharyngeal nerve is there, right? Glossopharyngeal nerve, which is in the tonsillar fossa. Here the treatment is tonsillar treatment. Okay, the site of action of polioviruses. The target site of action is motor nerve ending, which is similar to tetanus. That is, the poliovirus first enters into the anterior horn cells of spinal cord, and it spread into the muscle and causes muscle weakness and flaccid paralysis. Already I said no, AFP. The first thing you should take in mind treating polio is AFP, acute flaccid paralysis. In tetanus, spastic paralysis is seen. Okay, neuron degeneration is marked in polio. Okay, the neuron is infected with poliovirus, and the poliovirus causes degeneration of Nissl body. The Nissl body or aggravated ribosome in neural cytoplasm. Okay, the clinical manifestation. In clinical manifestation, the first thing you should take in your mind is incubation period. It's just like COVID vaccine, COVID virus, the 14 days. Okay, and in clinical manifestations there are 4 types. Okay, in apparent infection, abortive infection, non-paralytic infection, and paralytic stage. 91 to 96% of polio infected people are asymptomatic. Okay, that is in apparent infection, and the 5% people are symptomatic with mild infection. They have fever, malaise, sore throat, anorexia, myalgia, and headache. Okay, the 1% of people develops asymptomatic aseptic meningitis, that is non-paralytic poliomyelitis, and less than 1%. See, 91 to 96% of people is asymptomatic, 5% people is symptomatic, 1% people have aseptic meningitis, and less than 1% people only have this AFP, acute flaccid paralysis. Okay, yeah. The paralysis first affects mostly the proximal muscles than the distal muscles. Okay, here comes the very important part of polio. The paralysis starts at the hip, then extremities is first. Let me draw a line picture. The hip, and then spreads into the extremities. Okay, I will put a picture. Okay, don't worry. There will be a tripod sign. What is tripod sign? Child sits with flexed hip. Both arms are extended backwards for support. Okay, that is tripod sign. I will 1 minute. I will put a picture. Okay, don't worry. The tripod sign is very important here. Okay, the site involved of polio virus in human body is spinal, bulbar, bulbospinal, and cranial nerves. The spinal and cranial nerves are very important. That is sensory loss, no sensory loss. Okay, the polio virus causes biphasic course in children. Okay, first it causes aseptic meningitis, and then it causes, after the first recovery, okay, the child first manifests as aseptic meningitis, and it get cured. Again, the polio virus affect means fever with paralytic features will be presented. Okay, biphasic course is also very important. Okay, who are all more prone for polio virus are old children and adult people, and pregnant women. Trauma, tonsillectomy, and IM injections. Why tonsillectomy? I have mentioned over here. Neural spread. Treatment, tonsillectomy. Once the surgeon has resected the tonsil, the virus easily go into the glossopharyngeal nerve. That's why tonsillectomy people are more prone for virus, and IM injections. Okay, the lab diagnosis part has 3. Okay, that is viral isolation, antibody detection, and molecular method. First, viral isolation. We have to isolate the virus, right? So we want specimen. The specimen is collected like 3 duration, based on the duration. 3 weeks illness and 12-week illness. In 3-week illness, the sample is taken from throat. Okay, that is throat swab. And 12-week of illness, a rectal swab or stool sample. Okay, you people already know this is transmitted via the wait, let me show you. The fecal-oral route. Okay, so fecus, the sewage, you people already know. drainage with fecus is called, fecus is called sewage, without fecus is called sullage from SPM. Okay, this is not important over here. Okay, leave. Sewage testing. Sewage is tested for detection of polio virus under polio eradication program. Our government has implemented this. Okay, transmit is through refrigeration. And virus we need cell line culture. The cell line culture of primary monkey kidney cells, viral growth identification via cytopathetic effects in antigen detection and PCR. Okay, antigen detection is via IFT, immunofluorescence test, and PCR detects viral protein 1 region, which we have discussed in morphology part. Okay, antibody detection. The polio virus entered into our body. Our body produces antibody. This antibody is detecting for the detection of polio. Okay, a rise in antibody titer in the paired sera collected over 1 to 2 weeks. Here 1 to 2 weeks. Their 3-week sample is throat sample, and 4-week sample is rectal swab. Okay, 1 to 2 weeks interval in polio. Okay, antibody detection is via neutralization test. The neutralization test detects neutralizing antibody. The antibody infection. First, the antibody infection causes polio. First infection is polio produce strictly type specific response, and subsequent infection causes group specific response, common to all 3 types. Okay, if you want this part elaborate, just go and read. Okay, it's not much important. Okay, and the molecular method is via RT-PCR, which we people are doing, like viral protein 1 region primers, through which we are detecting wild polio virus and vaccine polio virus. Okay, this tabular column is very important. Okay, polio virus is available in 2 forms, injectable form and oral form. Okay, injectable form is like sulc, viral oral form is like bin. You remember like bun. Okay, bun we are eating through the oral mouth, so sabin is oral, sulc is injectable. Okay, the formulations of injectable polio vaccine is IPV. Injectable polio vaccine is of 3 serotypes, 1, 2, 3, and here let me come this part later. There are 2 doses, full dose injectable polio virus and fractional dose. Only the fraction of vaccine is given here. Oral polio virus is available in 3 forms here: trivalent, bivalent, monovalent. Serotype 1, 2, 3. Bivalent is 1, bivalent 2 and 3. Okay, you remember like this. Okay, already 2 is represented over here, so 1, 2, 3. And monovalent is any one serotype will be there in the oral polio. And national immunization schedule of India, we are giving it as 2 fractional doses, intradermal route, site is upper arm, 0.1 ml per dosage. First dose is given at 6th week of newborn, and second dose is given at 14th week of newborn. Here the 5 doses are given, 2 drops per dose oral. You people have already vaccinated for oral polio, right, in school days? Yeah. Bivalent OPV, the 5 doses are 0th at the time of the birth, that is considered 0th dose, and 1, 2, 3 doses are given at 6th, 10th, 14th week. 4th and 5th doses are booster doses, which are given at 16 to 24 month. Safety of oral polio in IPV is relatively safer than OPV. This is safer, but it does not provide safety for immunocompromised patient and pregnancy people and old age people. Efficacy is 90%, here 100%. Okay, Efficacy is decreased in other enteroviruses, diarrheal diseases, and breastfeeding. The efficacy is decreased here. Economy is, which is very expensive. This is cheaper. uh, Protection duration is short, here long lasting. Herd immunity here not provided, here provided. Local immunity is not stimulated, yes strongly stimulated. Prevention is, it prevents only the paralysis part. It prevents reinfection also. Stages, it does not require any stringent condition. It doesn't require any special conditions, okay, but it required special conditions. That is, refrigerator is important here. Minus 20 degrees Celsius, stabilized by magnesium chloride, acidic pH. What are the things? Refrigerator and magnesium chloride is required, and pH is also required here, but there is no any requirement here, okay. Vaccine associated poliomyelitis and vaccine derived polio virus, okay, there is no chance of vaccine associated poliomyelitis here. That's relatively more chance, okay. I hope this video is very useful for you. If you write these points, it will be much enough for an essay, okay. Thank you all.